What if the boundaries of life stretch far beyond what we've ever imagined? In this extraordinary account, a scientist claims to have studied beings from beyond Earth, creatures known as Exo-Biospheric Organisms, or EBOs. These entities, described with meticulous detail, blur the lines between biology and technology, evolution and engineering. From their streamlined genetics to their adaptations for a copper-rich, low-light world, their existence challenges everything we think we know about life itself. Are these beings the result of natural evolution on a distant planet? Or could they be the product of an ancient intelligence, mastering biology for a purpose still unknown? As you listen, keep an open mind, because whether fact or fiction, this story offers a fascinating glimpse into the possibilities of a universe teeming with life far more advanced than we've ever imagined. This is an analysis of the genetics and anatomy of Exo-Biospheric Organism, EBOs species D308. The findings suggest that EBOs share a common ancestry with our biosphere, as their genetics are based on DNA and are even compatible with our own cellular machinery. The EBO genome is highly condensed and uniform, with indications of artificiality and genetic engineering. The report also describes the gross anatomy of EBOs, including their skin, head, eyes, ears, brain, thorax, abdomen, pelvis, hands, feet, and endoskeleton. The biological systems of EBOs, including their respiratory, circulatory, excreto-sudoriferous, digestive, endocrine, immune, nervous, and musculoskeletal systems, are also analyzed. The report concludes with a discussion of the potential presence of artificial molecular machines in the EBO body. Introduction. EBOs are beings from an alternate biosphere, and their genetics and anatomy have been the subject of much study and speculation. This report presents an analysis of the genetics and anatomy of EBOs based on available data and observations. Genetics. First, genetics. Their genetics are like ours, based on DNA. This fact was very puzzling for me when I first learned about it. We imagine that beings from an alternate biosphere would have genetics based on a completely foreign biochemical system, and surprisingly, this is not the case. Several conclusions can be drawn from this surprising revelation. The one that immediately comes to mind is that our biosphere and theirs share a common ancestry. They're eukaryotes, which means their cells have nuclei containing genetic material, which suggests that their biosphere would have been separated from ours sometime after the appearance of this type of organism. The term exobiospheric organism is actually a misnomer, but as it's a historical term, it's still used. Their genetics are not only based on the same genetic system, but they're also even compatible with our own cellular machinery. This means that you can take a human gene and insert it into an EBO cell, and that gene will be translated into protein, and this of course works in reverse with a human gene inserted into an EBO cell. There are important differences in post-translational modifications that will make the final protein non-functional, but I'll discuss these later. Their genome consists of 16 circular chromosomes. You're probably familiar with the concept of intergenic region, or junk DNA. These are basically DNA sequences that don't code for proteins. These are evolutionary residues, transposons, inactivated genes, and so on. To give you an idea, in humans, intergenic regions represent approximately 99% of our genome. I'm aware that these sequences aren't completely useless. They can be used as histone anchors, as buffers to protect coding DNA from radiation, or even as alternative open reading frames, but that's rather peripheral. What's particularly striking about the EBO genome is the uniformity of these intergenic regions. We see the same sequences repeated everywhere, and the distance in BP between the genes is virtually the same throughout their genome. The result is a minimalist, highly condensed genome. In fact, it's much smaller than ours. Moreover, the quantity of protein-coding genes is even significantly lower than ours, probably due to genetic refinement, but also to biological processes that are absent in EBO. The uniformity of these sequences is a major indication of the artificiality of these beings. There is no complex organism on Earth that has such elegance in its sequences. There is no evolutionary pressure that can lead to this kind of characteristic other than genetic engineering. Speaking of genetic engineering, following sequencing of their genomes, we noticed a troubling and universal characteristic in the 5' of the regulatory sequence of each gene, which we call the tripalindromic region. The TPR are 134 bpi sequences containing, as its name suggests, 3 palindromes. In genetics, a palindrome is a DNA sequence that, when read in the same direction, gives the same sequence on both DNA strands. They serve both as a flag and as a binding site for proteins. The 3 palindromes in the TPR are distinct from one another and have been poetically named 5'P TPR, MTPR, and 3' TPR. The TPR is composed in 5'3' order of 5P TPR, 12BP chromosomal address, 12BP spacer MTPR, 12BP spacer gene address, 12PB spacer, and 3P TPR. The chromosomal address is composed of 4BP and is identical in each TPR of the same chromosome, but distinct between each of the 16 chromosomes of the genome. The gene address is a 64B+ sequence that is unique for each gene in the whole genome. It's therefore understandable that the TPR serves as a unique address not only for numerically identifying a gene, but also for identifying its chromosomal location. For those with only a basic knowledge of genetics, this is completely unheard of. No living thing in our biosphere has this kind of precise address in its genome. Once again, the presence of TPR cannot be explained by evolutionary pressure, but only by genetic engineering on a genomic scale. TPR opens the door to several possibilities. One of them suggests that EBO geneticists can insert or remove a gene from a cell in a way that is far more targeted and efficient than our technology allows. No proteins have been identified in the EBO genome that interacts with TPR. Rather, we believe that these proteins are exclusively targeted by external genetic engineering tools, probably used at the zygotic stage of embryonic development. The nature of these tools is unclear, but we definitely don't have anything like them. The probable absence of these proteins from the genome is a further indication of their artificiality. Given the high probability of artificiality of their genome and the apparent ease of modifying it with biomolecular tools, it's not out of the question that there could be polymorphism between individuals depending on their role and function. In other words, an individual could be genetically designed to have characteristics that give it an advantage in performing a given task, like soldier ants and worker ants in an anthill. Note that these previous statements are speculation. To my knowledge, only one individual genome has been sequenced. I can't make a definitive statement on genetic variation between individuals. I've talked a lot about intergenic regions. Now I'll briefly discuss intragenic sequences. Briefly, because there's not a lot less to say despite its obvious importance. Much like ours, their genes have silencers, enhancers, promoters, 5 UTRs, exons, introns, 3 UTRs, etc. There are many genes analogous to ours, which is not surprising given the compatibility of our cellular machinery. What's disturbing is that some genes correspond directly nucleotide by nucleotide with known human genes or even some animal genes. For these genes, there doesn't seem to be any artificial refinement, but rather a crude copying and pasting. Why they do it is nebulous and still subject to conjecture. There are also many genes which are not found in our biosphere, whose role has not been identified. Finding the purpose of these novel genes is one of the aims of the program. I'd like to note before going any further that this heterogeneity of genes of known and unknown origin is an undeniable proof of the artificiality of EBOs. To conclude with genetics, the mitochondrial genome, at the time I was working there, had not yet been sequenced. It's safe to assume that this genome would also be streamlined and possibly has some version of TPR. Transcription and translation, and protein expression. I briefly introduced the differences in post-translational modifications between human and EBO. This is hardly a surprise, as we often see the same thing between different terrestrial species. Obtaining a viable protein from a DNA sequence is a complex process involving hundreds of protein intermediates, each with a precise and essential role. A minor variation in this assembly line can lead to functional irregularities in the final product. So, it's no surprise that there are setbacks along the way when the first EBO gene transfection attempts failed to produce the desired functional protein in human cell lines. Fortunately for us, the work of what I imagine to be another team at another site has led to the development of an EBO cell line named EPIG11, derived from epithelial tissues. With this tool in our hands, we were able to transfect and overexpress proteins of interest in order to eventually purify and study them. For your information, we use a biological ballistics delivery system, aka gene gun, for our transfection needs, because other methods are not very effective with cells of this line. For example, the viral vectors tested cannot be internalized by EPIG11, and lipofection is too lethal. EPIG11, like most eukaryotic cell lines, enters a phase of exponential growth when exposed to fetal bovine serum. It's only half surprising that a cell line from such an exotic source should be sensitive to the growth factors present in FBS. In my opinion, this can be explained by the addition of animal genes to the genome, such as growth receptors. Gross anatomy. They are morphologically very similar to the gray aliens that are part of modern folklore. Their height is about 150 centimeters. They have 2 arms, 2 legs, and a head. Still, there are some notable differences. Skin. The gray skin that is often described in folklore is in fact a biosynthetic film, which likely serves to protect the EBO from a hostile environment. It doesn't provide effective protection against temperature changes, but it does offer adequate protection against the passage of liquids. It's possible that this film confers other advantages, but my knowledge on the subject is limited. Under the gray film, the epidermis is rather white, and the texture is very regular and without any hair. We do not see any defect other than the folds near the joints. It's described as greasy in one report, but that's not something I've observed. The same report states that a strong, lingering smell of burnt hair and ammonia is present when the film is removed. There are a lot of pores on the skin, crossing from the epidermis to a gland in the hypodermis. These glands and pores are the terminal part of the excretory sudoriferous system, which could explain the previously mentioned smell. Head. The head contains 2 large, oversized eyes, 2 nostrils without protuberance, a narrow mouth without lips, and 2 ear canals without auricles. There is a mandible, but the musculature is vestigial. There are no teeth or tongue in the oral cavity. The nasal cavity where the nostrils meet is compact and does not rise cranially but extends axially. There appears to be no equivalent to the olfactory bulb in the nasal cavity. The mouth leads directly to the esophagus and the nasal cavity to the trachea. The trachea and esophagus do not communicate. Eye. Like the skin, the eyes are covered with a semi-transparent biosynthetic film that offers the same environmental protection, while providing protection against certain wavelengths and light intensity. When the film is removed, a more traditional eye is revealed. It's about 3 times larger than a human eye, and there are no eyelids. The size of their eyes suggests they have excellent night vision. It seems paradoxical to cover them with a semi-opaque film. Perhaps they only need to wear it in a bright environment. Their sclera is the same color as their skin. The iris is pale gray, and the pupil is black and oversized. The lens is rounder than a human, and the musculature used to adjust focus is more developed. On the retina, there are at least 6 types of cone cells. The responsiveness of each of these 6 types of cone is specific to a wavelength band, with a minimum of overlap between each other. The result is a broader visible spectrum. Ear. As mentioned, the outer ear has no auricle, and the ear canal is unremarkable. The inner ear has all the characteristics of a typical vestibular and cochlear system, although the curvature of the cochlea is more pronounced than a human. This probably results in greater hearing acuity for low frequencies. Brain. The brain is tetraspheric, i.e. composed of 4 major sections. The sections are separated by transverse and longitudinal fissures and are connected to the central lobe, which acts as brainstem and cerebellum. The volume of the brain is around 20% superior to that of a man of the same height. It has a much more pronounced level of gyrication than an average human. Moreover, the ratio of glial cells to neurons is also slightly higher than in humans. It is important to mention the presence of nodules on the central lobe. Histological analysis of these structures reveals a kind of intricate biological circuitry. It is speculated that these nodules are essential to interact with their technology. Consequently, determining the proteome of these structures is an absolute priority for the program. Neck. The neck is proportionally longer than that of a human, and at the same time relatively thin. As mentioned, the esophagus and trachea are separate. There are no vocal cords in this region. Thorax. The musculature of the thorax is underdeveloped. Muscles equivalent to the pectoralis major can be seen. We can also see the trapezius and deltoid muscles. The sternocleidomastoids are well defined. The ribs and sternum are clearly visible. There are no nipples. The abdomen is wider than the thorax and bulges slightly forward. There is no navel. Pelvis. The pelvic bones are apparent. There are no genitals or anus. Hands and feet. Their hands have 4 digits, including an opposable thumb on the medial side. They have no nails, and the texture of their fingerprints is composed of concentric circles. Fingers are proportionally much longer than in humans. Unlike humans, finger musculature is entirely intrinsic to the hand. In other words, the muscles used to move the fingers are not in the forearms but entirely located in the hands. At first glance, the feet consist of just 2 digits, but a necropsy soon determined that each toe was made of 2 fused digits. The medial toe is marginally longer than the distal toe. The feet are relatively longer and narrower than in a human. Their musculature, however, is vestigial. The EBO's endoskeleton is very similar to ours, at least in terms of composition. There's collagen, hydroxyapatite, but also copper oxide crystals where marrow would normally be found. The role of these crystals has not been established, but it is not a crystallopathic condition. The blood cells of the myeloid lineage, or the equivalent for these creatures, therefore mature in a different location than in humans, i.e. in the thymus-like organ. A transverse section of the bone reveals osteon and osteocytes. There appear to be few osteoblasts and no osteoclasts. This indicates that the bones are no longer growing and cannot absorb the minerals present or adapt mechanically to changes in posture. Biological system. Respiratory system. Their cellular respiration is equivalent to ours, i.e., they need to oxidize organic components to produce energy. Their lungs have no reciprocating action, but rather have a unidirectional flow of air, similar to those seen in birds, which is more efficient than ours. It is speculated that this is in response to the brain's elevated metabolic needs. Vocalization is produced by vibration of the wall membrane at the junction between the 2 air sacs. The circulatory system. The circulatory system of EBOs is rather analogous to ours. The heart is located in the mediastinum, but in a more medial position, directly beneath the sternum. The heart has 2 ventricles and 2 atria. There is an aorta, a pulmonary vein, a pulmonary artery, and a vena cava. Blood flowing to the pulmonary capillaries via the pulmonary artery is pumped against the flow of air, maximizing gas exchange efficiency. The blood-gas barrier is relatively narrow in these capillaries, at least compared to a human. Then oxygen-rich blood is returned to the heart and then expelled into the aorta and the rest of the body. Before returning to the heart, the blood will pass through the hepatorenal organ, which, among other things, filters and controls osmotic pressure of the blood. The blood itself is also analogous to that of a human. However, the proportion of plasma is much higher. Albumin is in similar proportion. Hormone levels are much lower. Metal ion levels are much higher, particularly copper, and glucose levels are significantly higher. The color of the blood is brownish, given the higher proportion of plasma and concentration of metal ions. On the cellular side, there are erythrocytes which, in addition to hemoglobin for binding oxygen, display several complexes capable of binding copper ions. It's not clear what role these copper ions play, but we believe it neutralizes blood ammonia, among other things. Several cell types with leukocyte characteristics have been observed, but no comprehensive knowledge of them exists. Platelets are present, but in smaller proportions than in humans. Excretosudoriferous system. This system is completely different from what I've seen. As mentioned earlier, there is no large orifice, like an anus or urethra, to get rid of biological waste. Instead, there are countless small pores on the surface of the skin. There's a large medial organ called the hepatorenal organ, which acts as both kidney and liver and is central to maintaining homeostasis. This organ is highly vascularized, and the blood must pass through it before returning to the heart. Its role is, among other things, to purify the blood of metabolic waste. Waste is excreted into the equivalent of a ureter, which branches out into 4. Each branch flows towards one of the 4 limbs, and in turn these branches divide until they end up as thousands of excretory pores. The motility of this excretory system is mediated by a weak peristalsis at the proximal level and on the 4 main branches. Peristalsis ceases around the first distal junction. As there is no urea cycle, the ammonia concentration at the exit of the hepatorenal organ is very high. This ammonia is carried to the pores and gives the distinct odor I mentioned earlier. The rationale behind this unusual excretory system is directly related to this excreted ammonia, which enables thermoregulation by evaporating on the skin's surface. The greater the physical effort, the greater the metabolism. This in turn leads to a rise in temperature and a corresponding increase in metabolic waste via amino acid catabolism. This leads to an increase in filtration and ammonia excretion, which ultimately lowers body temperature. Digestive system. The digestive system is extremely underdeveloped. There is no stomach in the familiar sense. However, there is a pseudostomach located at the transition between the thoracic and abdominal cavities. This organ is not involved in digestion, but only serves as a reservoir. A sphincter controls the flow of food into the intestine. The intestine is limited to the equivalent of our small intestine, i.e., it only serves to absorb liquids and nutrients and acts as the main digestion site. It has villi and microvilli like ours. The intestine ends in the hepatorenal organ, where non-digested matter is transported to the ureter and excretory system. Residues are dissolved in the ammonia of metabolic waste for excretion. There's an organ near the pseudostomacal sphincter that secretes digestive enzymes directly into the intestine. This organ is inspirationally called the digestive organ. It secretes mainly proteolytic enzymes and glycoside hydrolases. Given the absence of teeth, the narrowness and rigidity of the esophagus, the absence of a true stomach, and the absence of defecation, it is strongly believed that EBOs can only consume food in liquid form. It is assumed that, given the high metabolic needs of their brains, this food would have a high carbohydrate concentration. In order to meet other metabolic needs, there must also be a high protein content in the food consumed. These two statements are supported by the type of enzyme secreted by the digestive organ. It is therefore speculated that the food consumed is a sort of broth rich in sugar and protein, which probably also has a high copper content. Given the strict limitations on the type of food that they can consume, it's unlikely that this type of creature could survive in our biosphere without technological support. Endocrine system. Knowledge of the endocrine system is minimal. We know that cells are receptive to bovine growth hormones, so it's assumed that certain functions are regulated by such a system. Endocrine mechanisms are very complex, and it goes without saying that they are best studied on living subjects. Immune system. The immune system is another unknown. There seems to be an innate immune system, but there doesn't seem to be any adaptive immunity, at least not similar to what is known. There's a thymus-like organ near the heart that's proportionally larger than in humans. This organ seems to be where all blood cells mature. Some cells have leukocyte characteristics such as granularity. The immune cells that germinate here have a high copper concentration. The surface receptors of innate immune cells have not yet been characterized, so we might as well say that all the work remains to be done. Nervous system. The nervous system is also relatively similar. The spinal cord begins at the base of the central lobe of the brain and propagates down the vertebral column. In the vertebrae, there are ganglia made of afferent and efferent neurons. In short, other than the CNS, there is nothing out of the ordinary. Musculoskeletal system. The musculoskeletal system is very ordinary, albeit underdeveloped. Most of the human skeletal muscles have an equivalent. Only the hands, feet, and forearms are different. It should be noted that the proportion of type 1 and type 2 muscle fibers is different from that in a human. Indeed, type 1 outnumbers type 2 by about a factor of 10. Artificial system. We speculate that artificial molecular machines may be present in the body, and that copper, if present, would be essential to their function or assembly. Importantly, no AMMs have been observed. Conclusion. Unlocking the paradigm, reconsidering the EBO narrative. The article on species D308 presents a challenge not only to our scientific understanding, but also to our paradigms of biology and evolution. While critics dismiss it as speculative, black swan phenomena have historically shattered deeply entrenched assumptions, forcing a re-evaluation of long-held theories. If the claims are true, we must adapt our frameworks to incorporate a broader cosmic perspective. The skepticism surrounding the article stems largely from paradigm lock-in, the tendency to view phenomena solely through Earth-centric theories. Our science is shaped by terrestrial observations, making concepts like alternate biochemistries or engineered organisms appear implausible. Discoveries of extremophiles thriving in Earth's harshest environments, or evidence of exoplanets with unexpected compositions, remind us that nature often defies expectations. If EBOs are real, their biology could represent such a black swan event, expanding our understanding of life in the universe. Our evolutionary biology assumes gradual natural adaptations. EBOs challenge this with traits suggesting engineered evolution and co-adaptation with advanced technologies, potentially by an ancient, spacefaring species. On the species' origins, habitat, and copper-rich, low-light environments, the EBO's physiology aligns with a planet orbiting a dim star, where photosynthesis would favor pigments like rhodopsins, producing ecosystems dominated by purple or blue plants. Herbivorous prey, potentially large arthropod-like creatures, would bioaccumulate copper and sugars, explaining the EBO's dietary reliance on liquid, high-protein, and copper-rich fluids. The EBO's adaptations suggest an efficient predator in a non-violent niche. Their large eyes, night vision, and liquid diet imply stealthy or opportunistic feeding rather than violent predation. Ammonia-based thermoregulation supports bursts of high-speed activity in the pursuit of prey, consistent with a highly competitive environment. The described traits, streamlined genome, reliance on liquid diets, and artificial enhancements, may reflect evolutionary pressures of spacefaring life. Coevolution with advanced tools and AI could reduce the need for complex digestion or immune systems, as technological environments mitigate risks and could indicate technological symbiosis. Streamlined biology could result from deliberate genetic engineering to optimize efficiency for specific roles or habitats, particularly in interstellar travel. If real, EBOs likely represent an ancient advanced species far older than humanity. The presence of biological circuitry in the brain suggests a species intertwined with technology, possibly technological coevolution, and using their biology as an interface for advanced systems. The minimalist genome could indicate reliance on external systems to supplement biological limitations, likely a hallmark of advanced civilizations. Task-specific genetic polymorphism suggests mastery of artificial evolution, with individuals engineered for distinct purposes, much like worker or soldier ants in a colony. Their adaptations might reflect long-term survival strategies for interstellar environments, with engineered resilience to diverse ecological niches. Space, the final, unlimited frontier, a cosmic perspective. Humanity often views evolution and survival through a terrestrial lens, underestimating the profound challenges of long-term space exploration. But the EBO narrative challenges us to consider what adaptations are needed in an infinite frontier. Space environments demand efficiency, resilience, and adaptability, traits reflected in the EBO's biology. Advanced species may bypass traditional evolution, using genetic engineering to survive and thrive across the universe. The described biology implies a rich ecological context where EBOs evolved, possibly on a resource-rich planet or as part of a broader interstellar ecosystem. But their traits might also indicate the influence of a larger spacefaring community where biological and artificial entities coexist in a more evolved interstellar ecology. Closing thought, embracing possibilities. The species D308 narrative may indeed challenge our scientific norms, but dismissing it outright risks overlooking a paradigm-shifting discovery. Whether speculative or a glimpse into a cosmic reality, the story compels us to ask, what is the nature of life in the universe? How does technology reshape evolution? Are we prepared to expand our understanding of biology beyond Earth? Space is the ultimate frontier, not only for exploration, but also for expanding the limits of our imagination and science. Whether or not the EBOs exist, the concepts presented demand that we remain open to possibilities far beyond our current understanding. Only then can we truly grasp our place in the cosmos. Summarized and read by J.H. Thanks for listening.